right, thank you very much for the warm welcome, and I would like to continue with saying thank you for being present today to listen to our capstones. It's really exciting for us to be here and to have you here. So my name is Mohamed Saleh, and I'm going to be presenting the Inflammasome Gene Expression Analysis in Induced Pluripotent stem cells. And my supervisor is Roxana Zakharian, and my advisors are Lana Karapetyan and Gisane Khachatyan. Unfortunately, they're not here today. So to begin with the simple introduction, the idea is to analyze the gene expression of a very common disease called the FMF, which stands for the familial Mediterranean fever, which is an autoinflammatory rare monogenic disorder. Another thing that we'll need for this presentation is the induced pluripotent stem cells, or iPSCs, which are basically cells that we are getting by inducing stem cells. We'll see more about that in next slides. And the third cornerstone is the inflammasome gene expression, which basically is going to check for us the signaling of the protein synthesis or protein complexes, as well as the genetical sequence. And last but not least, basically what are PBMCs, which stands for peripheral mononuclear blood cells. So to begin with, familial Mediterranean fever is a recessive disorder, meaning that the affected person will have to carry both genes from both parents in order to have this disease. But sometimes carrier people do exhibit some disorders of FMF. Moving on, we have these mutations usually in two hotspots. The main one is the 10th exon, but there are also existing some mutations in the second one. The most common mutations being the M694V and the M694I. And basically the current state of the problem is that there's a large population coverage, about 37 to 39% in Armenians and Iraqi Jews, as well as 20% in Turks, North African Jews, Ashkenazis, and the Arab populations. When you look at these numbers, they might seem small, but when you look at the populations they represent, the numbers talk large. And the problem is that there's a huge lack of data and experimentation regarding the gene expression. There is enough data about the types of mutations, but there is no enough comparison between the control patients or the healthy patients, as well as the patients that do exhibit the FMF disorder. And the existing medications are basically colchicine and IL-beta inhibitors, which we'll see about here. So to explain the process simply in terms of the medications available now, we have a mutation. It's not visible, I guess, here in the gene, which causes the pyrine protein, which is the main protein responsible for producing the inflammasome reaction or deformation, basically becomes mutated, does not form well in the quaternary stage of the protein folding, which results in creation of inflammasomes without any reason, which causes to inflammation and basically stuff like fever, cirrhosis, and joint pain. So when we apply colchicine, we basically stop these inflammasomes. You can imagine it as something like an antihistamine. Meanwhile, when we use the IL-beta receptor blocking, we basically do not allow the cells to receive that signal or receive that inflammasome output. And now you might ask, why would we use iPSCs for such a study for doing genetics? Well, the main reason is that there have been many successful cases where generation of iPSCs has allowed thorough study of the gene sequencing as well as the behavior of the cells. So not only will we be able to examine the genetical differences, but also will be able to, in the future, understand the exact behavior of these cells. And mainly iPSCs are being used in drug screening and, most importantly, gene modification. There are two approaches for it. The classical approach, which is basically to apply the Yamanaka factors, and the more advanced technologies, which is basically applying plasmids, which is basically inserting the new DNA or the alteration of DNA directly into the nucleotides. And now for the PBMCs, or the peripheral blood mononuclear blood cells, basically the reason why we need those cells is because they're easy to extract. We can extract them from the patient's blood and immediately be able to induce them into pluripotent stem cells. And not only that, we can also apply on them the DNA or the genetic testings. And another even good thing, since we're studying inflammasomes, mononuclear blood cells are those basically inflammatory cells, or to call it in a better way, they're the cells that are responsible for the second immune response. The main two being the neutrophils and the macrophages. And for the inflammasome expression, since we're targeting specific genes and not the entire 16th gene, we will basically be targeting the already well-known hotspots for the FMF disorder, which is the MEFV gene, CASP1, and the P65, which is responsible for the formation of the pyrene protein. And we will use the GAPDH as basically a reference background in order to understand how different the expression of the genes are. Moving on, the aim is basically to provide a proof of concept that we can identify which genes are expressed higher, so that in the future we can basically apply gene editing techniques in order to completely turn off this, let's say, the bad gene, so that we can alter the behavior for the somatic cells, meaning that basically we're curing these patients from the FMF disorder. And what we are expecting is to provide a thorough proof of concept, a validation of the existing methods and data, and of course providing more data since there's a lack of it, and of course minimizing the usage of the medication. Now for the fun part, the methodology. So for the PBMC's extraction, it's rather simple. We basically we take blood samples from people who are healthy and from people who have FMF, and we verify that using simple PCR kits that do show the expression of the specific genes that we've already mentioned. And then we use blood processing buffers such as fecal, which will be used to divide the two type of blood cells that we'll have, which are the granulocytes and red blood cells, as well as the mononuclear cells. Then we'll centrifuge it and we'll see how will that work in a while. And then we'll basically wash the cells to ensure that they are sterile, and then preserve them in liquid nitrogen until we conduct the experiments. As you can see here, I've extracted my own blood for this experiment, so I can say that I've put my blood, sweat, and tears into this project. And as you can see here, the white substance is the fecal, which will be used to later divide the blood. I will show an illustration for that for my visual people. Here we can see the centrifuge, which basically will be spinning the blood around 400 G, which is I think approximately 1,500 rounds per minute. It varies depending on the machine. And here we can see the visual difference before and after the centrifugation. Basically we have here the things that we don't need, which is the red blood cells and the plasma. And here, this tiny layer is where all of the mononuclear blood cells exist. And that's what we basically will be extracting. And then for the iPSCs, unfortunately, due to time constraints as well as how critical it is to induce them ourselves, we basically have to source them, verify that they are either control or FMF patients, again using standard PCR, and then extracting the coding DNA or the cDNA from them. And then for the main part of this research, which is the real-time quantitative PCR, which will basically show us in real time, based on the number of cycles for a PCR, what is the amount of fluorescency, or basically how significant is the expression of the gene. In order to do so, we'll have to prepare a specific mix that will be containing fluorescent dye, as well as the basic PCR kit for 20 ampules, because we'll have to be testing 10 primers that we've already mentioned for the specific gene hotspots, for a control sample and for a patient with FMF, and then divide the cDNA into the 20 ampules, add the primers, and then basically centrifuge and make sure that everything's mixed, and then place it into the rotor gene queue and wait. Now, here we can see my 20 ampules. Here were the cDNAs, basically transmitted into all. Here we can see the master mix, which is basically composed of distilled water, the primers, the cDNA, and of course the qPCR main test. And over here to the right, you can see that every row is basically having the forward and the backward primer. And then for the actual machine, or how does it work, it basically heats up for 5 minutes to 95 degrees to assure that every single DNA strand has been denaturalized, meaning that the strands have been broken up so that we can apply the polymerase chain reaction. And then the cycle begins for 15 seconds, maintaining that temperature. Then it drops to a temperature called the annealing temperature, which is around 60 degrees Celsius for 30 seconds. And then the temperature increases back to ensure that the extension has been completed. And then steps 2 to 4 are repeated 40 times. And here's the machine, so let's wait for it to generate the results. Great. So here we can see a very simple graph, yet it's very informative. The reason is, well, let me explain the axis first. On the x-axis, we have the number of cycles. Basically, as I mentioned, it stops at 40, because after that, we basically have a decrease in the amount of DNA copies, and anything before that, we won't get the exact graph. And on the y-axis, we have the normalized fluorescency. Basically, the higher the value, it means the more the gene is expressed. And over here to the right, we can see the pink slots from 1 to 10 are basically for the FMF patient, the 10 ampules that we've mentioned earlier. And then from 11 all the way to 20, even though it's here till 16, it's just not visible on this picture, is basically for the controlled patient. And then we apply something called the CT, which is basically to see at which specific cycle do we have the maximum fluorescency, because that's what we need for the gene expression. And then we'll calculate the simple difference, or delta CT, which is basically the difference of the expressed gene versus the background that we're using as control. And then we'll do the second derivative, or delta CT, which is basically seeing the difference between the FMF patient and the control patient, and then raise that value to the power of 2 to understand by how many folds, since DNA is in copies of 2, we have 2 strands, and how many folds is the gene different. As we can see here, we have the main values that are above 2, which is the international standard for real-time PCR. Any values higher than 2 is called an upregulation, which means that the values are significant. Anything lower than that is considered insignificant. And as we can see here, even though these are all of the gene citations that the literature has proposed to view, we only see significance in 3 of them, which is basically the MEFV gene, the pyrene related gene, the P65, and the NIRP, which indicates that we do not have to change every single FMF gene that we know in order to cure the disease. And basically, these patterns suggest that the inflammatory response is indeed altered, but it should be only interpreted as preliminary, as this was conducted to provide a proof of concept and not to provide a statistical, basically, proof, even though these values are statistically significant based on the PCR standards. And that was all. Thank you very much for listening. Any questions?
Yeah, what was the engineering part of it?
Great question. So the engineering part of it was basically to find this, how do I call this? Find this framework where we can do the testing and get a very minimal result, not doing basically data science or statistics, and to understand basically if we can apply the techniques of genetical engineering or not. So for this specific study, the main part of engineering was basically the tissue engineering, which is generating the induced pluripotent stem cells, and basically applying the methods of biotechnology and also biomedical imaging. But unfortunately, the images were not very significant, therefore they weren't applied into the presentation, but they are included in the paper.
Your original draft had Dr. Arsen Harakyan as an advisor. Yes, it's just a bit finished.
Yeah, unfortunately, there were some technical issues that had to change.
Do you see any ethics involved in your work?
Definitely, the extraction of the FMF, basically the blood samples from both the controlled patients and the patients with FMF had to be both licensed by the Armenian government, and also they had to basically, they gave us their consent to take this blood for the research. And another thing is that there could be an ethical crisis, meaning like people would be really afraid to test their blood because like what if everyone has FMF? But in the same time, this specific experiment was done ethically, and we do have the ethical statement in the paper.
Okay, so you apply for ethical clearance and
Definitely.
Okay, good. That's good.
What is the sample size that's required to prove statistically that this is correct? Have you estimated the number?
Well, to prove if it's statistically significant or no, we'll at least need 3 different samples from each patient, and based on the previous, based on the literature review, from 30 to 50 patients, 3 samples from each is enough to prove that something is statistically significant in a population. So, for example, in Armenians living in Yerevan, for example. But then the same experiment will have to be conducted in multiple populations to say that it is generally significant. Have I answered your question?
Yeah, and for example, for Armenians from Iraq and from different, is it dependent from the ethnicity, or it is different from what, or it is like globally the same sample size? How is defined in your case?
Well, usually for the sample size, the larger it is, the better. But the idea is that if the sample has the same mutation, it is more significant than if it has multiple variations. So, for example, in the Armenian populations, usually the mutations are the M694V or I. So, since they are all of the same type of mutation, the significance is higher. But if we compare, for example, Armenians living in Yerevan versus Armenians living in Iraq, their mutations might be probably different since their heredity is also different. So this is a study basically on epigenetics, basically on their ancestry and how did they get basically their FMF from, or where did they get it from.
Yes. If a student in the future, let's say next year, is going to continue your work somehow, what sort of future work or direction for next steps might you suggest?
Oh, definitely. The most basically evident one will be to, in the case of data science, to provide a more significant result in terms of biostatistics. In terms of engineering, it will be basically to apply the concept of the gene editing techniques and to show the next part of the proof of concept, which is that after changing these nucleotides, we will have healthy somatic cells. So it's more of a tissue engineering project than a biotechnology project.
How long you are engaged in the project, in this project? How long?
Since I've basically started it in last September, if I'm not mistaken, so about 8 months.
Last September.
Yeah.
So I'm just interested. Do you think that without the proper background, because we don't have such core courses, right, in the program?
We do not have core courses, but we do have distribution requirements and free electives that are heavily related to this. For example, I've taken personally tissue engineering, biomedical imaging, and introduction to bioscience. And the reason why I took these courses was that I will be able to make a capstone that's related to that. Thank you. All right, if that's all of the questions. Any questions from the audience?
If supervisor wants to.
Oh, of course.
Hi everyone. So explain with my previous season. You came of course introduction to bioscience, and he expressed interest to only in this field, and he definitely has some idea what to do. I want to work with stem cells, and it's kind of not the usual or common topic because those technologies are just developing in Armenia, but we talk had laboratory at the Institute of Molecular Biology, and they had all the facilities. He really easily engaged in all the work and did everything. The main part he has done by himself, and his motivation and knowledge from disciplines related to engineering, health science really helped to make it successful. So I wish you good luck, and I hope that you will still have your career development this track visible for all of us in for faculty and also community members.
Thank you very much. All right, if that's it, then I will leave the stage for the next presenter.